Good morning, everyone, and thanks for the opportunity to present this overview of tuberculosis vaccines. I have no conflict of interest to declare. There has recently been quite some excitement about tuberculosis vaccines, both from clinical studies, as you can see here on the left, and from more basic science, as you can see from the right. I will discuss them later on. For now, what I'll show you here is what I'm going to talk about. I'm going to talk about the need for new tuberculosis vaccines, about barriers to developing those new tuberculosis vaccines, about the current TB vaccine clinical development pipeline, about recent developments in discovery, and then finally a few words about implementation. So let's start with the need for new tuberculosis vaccines. So what you see here is a summary of the TB data from the latest WHO Global Tuberculosis Report. They're about 2019. As you can see, the World Health Organization estimates that in that year, 10 million people worldwide fell ill with tuberculosis, leading to 1.4 million tuberculosis deaths. The majority of these TB cases are in the two WHO regions, basically around here, around Bangkok. Southeast Asia and Western Pacific together account for more than 60% of all the cases, and 25% of the cases occur in Africa. And what you can see from the little map here on the left is the incidence rates, and you can see that the highest incidence rates are in sub-Saharan Africa, but also pretty high incidence rates in South and Southeast Asia. Clearly, TB HIV co-infection is still a major problem. The WHO estimates that in 2019, there were over 800,000 people living with HIV who got tuberculosis, over 200,000 TB deaths among people living with HIV. The majority of the TB HIV cases are in Africa, about 3/4 of them, and almost 15% in Southeast Asia. And here you can see again on the map, you see here the proportion of notified tuberculosis patients who have HIV infection. And you see that is definitely highest in Southern Africa, but also pretty high in, for example, parts of the former Soviet Union. There is a vaccine, there has been for a very long time a vaccine against tuberculosis. It's called BCG, which stands for Bacille calmette guérin. It's a live attenuated strain of Mycobacterium bovis. It was actually developed in the 1920s and 1930s, has been in use since the 1930s. And what you see here or in the graph is a meta-analysis of randomized controlled trials that have been done up to 1988. And what it basically shows you, and you can see here so this is the relative risk, and the further the relative risk goes away from 1 to the left, the higher the vaccine efficacy. And you basically see that the vaccine efficacy in these trials is all over the place. It ranges from 0% to 80%. People have been doing over the years a lot of work to try and understand what this heterogeneity is due to. And this is from this same overview, which is actually a meta-regression of those various trials and other studies. And that clearly showed that vaccine efficacy for BCG is associated with higher latitude, meaning that the farther you get away from the equator, the better it works, that it is associated with neonatal age and with higher quality studies. And the overall conclusion is that BCG does offer protection in neonates, in particular against meningitis and miliary tuberculosis, which are the main causes of TB-related death at that age. And that for that reason, it is the worldwide use for neonatal immunization, at least in most of the countries, at least in those with high TB incidence, should definitely be continued. However, it has a questionable efficacy in adolescents and adults, and it has compromised safety in those who have cellular immune deficiency, including HIV, because of its live attenuated nature. And this is basically to show that we do need something new. This is the basically the levels of achievement of the targets for the End TB strategy, which is WHO's strategy to eliminate tuberculosis by the year 2030. And it tells us where we stand on the three main targets in 2020. And you can see that for TB incidence rate, the target was a reduction of 20% worldwide. We've only reached 9%, For the number of tuberculosis deaths, target 35% only reached 14%. And the percentage of people with TB facing catastrophic cost target was 0% by now, still looking at 49%. Meaning we are definitely not yet there and the pace is too slow. And what this graph here shows you is the estimated tuberculosis incidence. All tuberculosis cases is notification, but this is what we should be looking at here. And it shows you how far we're still off from the target for 2030, the elimination target, which is here. So there is still a huge gap to be covered. And already earlier on, modeling work has showed, and this is what is here shown in this arrow here in the middle, that in order to reach elimination, we need to have such a drop-off in tuberculosis incidence and number of deaths that we definitely need new tools, and a new vaccine is probably the essential one here. This is why the WHO has, over the past few years, defined a number of product profile characteristics for the new tuberculosis vaccines. So this is basically saying this is the kind of vaccine that we need, and this is the minimum characteristics it should have. And these 3 PPCs, as they're called, are from left to right: a safe, effective, and affordable TB vaccine for adolescents and adults. This is definitely the most important one because this is going to have the impact on the epidemic. The second is an affordable TB vaccine for neonates and infants with improved safety and efficacy compared to BCG. And the third, and that's really rather new thinking, is a therapeutic vaccine to improve tuberculosis treatment outcomes. If we look at the one here on the left, here really the aim is to prevent pulmonary tuberculosis disease, because that is also, it's the most important form of the disease clinically, but it's also the one that is infectious. And the desired characteristics here are a 50 or greater % vaccine efficacy with a duration of 10 years or more in subjects, both subjects with and subjects without evidence of already existing latent mycobacterial tuberculosis infection, with a favorable safety profile, amongst others safe in people living with HIV, other immunocompromised people and elderly and so on, with a minimum number of doses and boosters needed, and the price that would not be a barrier to access, including in low and middle income countries. For the childhood vaccine, it would really be about prevention of TB disease, including severe disseminated tuberculosis, so that's miliary TB, TB meningitis and pulmonary TB, with an 80% or greater efficacy, all superior to BCG. Again, duration of 10 years or more. Here, co-administration with other vaccines in the expanded program on immunization is obviously very important. The vaccine should have improved safety compared to BCG. Again, the minimum number of doses and boosters needed, no interference with other EPI vaccines, and price not being a barrier. And then finally, for the therapeutic vaccine, the idea is here to have a vaccine that would either prevent recurrence after cure, so that's a patient who's been treated successfully for tuberculosis, but we know that they look at 2 to 5% risk of relapse or recurrence, and the vaccine should prevent that. Or the vaccine should increase the cure rate when given during treatment, for example, in people who are for some reason hard to treat. And obviously, drug-resistant tuberculosis would be an example. A therapeutic vaccine should be effective in all TB treatment, both drug-sensitive and drug-resistance. The desired vaccine efficacy would be during the 1 year of follow-up. Clearly, in this case, one would follow up for a shorter period. 50% of greater, again with favorable safety profile, minimum number of doses, and price not being a barrier. Now, in the second part of my presentation, I wanted to talk a little bit about the major barriers that currently exist to developing new TB vaccines. And for doing that, I would like to start with just quickly going over the TB vaccine development pathway, which is not very much different from any vaccine development pathway, but it's just sort of to synchronize our watches on the various phases in the pathway. So it starts with discovery, and this is the phase in which antigens that are immunogenic are identified, in which platforms are identified and developed, in which adjuvants are identified. So basically, this is where the engineering of the vaccine is done. Studies here are proof of concept studies, clearly animal studies, to see whether these candidate vaccines would have any efficacy at a biological level. The next stage is the preclinical stage, in which especially the manufacturing and the quality control process, so making sure that one has a product that can be reproducibly evaluated, that product is available. That is very important. So it basically prepares for phase 1, also looks at dose ratios. And again, here animal studies are very important. And then if a product passes that stage, candidate, it goes into clinical development. And here the stages are classically 1, 2A, 2B, and 3. And in phase 1 is the first in human phase, which is primarily looking at safety in healthy volunteers, but also to some extent immunogenicity. If that is successful, a candidate can move on to 2A. And from 2A, basically studies are done in the population of intended use. So if we're looking for a vaccine that should be used in the elderly, one should look in the elderly from phase 2A onward. So this is really about dose finding, phase 2A, safety and tolerability again, and immunogenicity. From phase 2B onwards, in addition to the other characteristics that are evaluated, clinical efficacy becomes important. Usually, if phase 2B are relatively small studies that can give an indication of clinical efficacy, and then obviously the phase 3 studies that are meant to complete a dossier for licensure, for registration, is really about clinical efficacy at large scale and safety and tolerability. And then after introduction, there is a lot more that generally needs to be done. For example, safety surveillance, pharmacovigilance, effectiveness in the real-life world, barriers to vaccine implementation, impact, the public health impact of these vaccines, cost effectiveness, and so on. So this is the entire pathway in terms of the research that needs to be done. Now, if we look at those barriers, I have identified 5. The first one is really in the discovery space. And that barrier is that we have very limited knowledge of the human protective immune responses that are needed to prevent tuberculosis disease. What we know by now is that conventional T-cell responses are necessary for protection, but they're not sufficient. We need something in addition to that, and we basically don't know exactly what that is. And if you look at this little graph here, this is the sort of current paradigm as it is emerging about the natural history of tuberculosis. So one gets infected, then can eliminate that infection in early stage, but can move on also in early stage quickly on to having tuberculosis, or have latent TB infection that can remain for a very long time, also in later stage be eliminated, or lead to rapid or less rapid, slow reactivation of disease. So all these various stages. Now, basically what we don't know is what drives the transitions along this natural course of infection, and what drives clearance and what drives this move towards disease. We basically don't know what the key immunological parameters are here. We also don't know what the role is of non-conventional cellular immunity, of antibody responses, and of trained innate immunity. Both antibody response and trained innate immunity are getting a lot of attention at the moment, and they seem to be much more important than we previously thought. And then a third important element is how to direct immune responses to the lungs, because that's where these vaccines should be active. So if a vaccine is given intradermally, how do we make sure that it's actually at the level of the lung parenchyma that they exert their protection? A second barrier has to do with the animal models. Basically, we don't know whether our animal models really predict prediction in humans. And what you can see here is the 3 animal models that are used most. It's the mouse, it's the guinea pig, and it's non-human primates. And the further we go to the right, the more we think that the immune responses mimic those in humans. But we're not entirely sure. So as a result, the correlation of what we predict in terms of protection of a vaccine candidate in animals and humans is limited. It has to do with the animal biology. It has to do even in, for example, non-human primates, quite some variability in outcomes. So animals responds pretty different to challenges with Mycobacterium tuberculosis. It responds pretty different to candidate vaccines, and we don't know exactly why. Another thing that we don't know exactly yet is what an infectious challenge or an infectious dose looks like. Is that something that's one-off? Is it something that needs to be given over a prolonged period of time in very small doses? So what is actually the natural biological way in which humans get infected, and how can we mimic that best in animal models? And the problem here is really that because there have been so few studies, clinical studies this far, that have shown protection signals of vaccine candidates, it's been very hard to validate these animal models. And we're only starting to do that now that there are studies that are showing a protection signal. The third problem, I think, is really key, and that is that there is a lack of agreed laboratory correlates of protection. And I want to illustrate that by the by now notorious example of the BCG MVA-85A boost combination. So this was a pretty promising vaccine candidate. So it was giving this affected vaccine with the 85A antigen as a boost in people who had been primed with BCG. And actually, in studies in non-human primates, and what you should be looking at is here, BMVA, it actually looked quite promising. This is the lung pathology. This is the bacterial load in the lung in animals that were vaccinated by these various vaccine candidates and then challenged. And you can see that the MVA 85A boost performs pretty well in both cases compared to BCG and compared to non-vaccination. And in human studies, these were phase 1 studies, there was actually quite nice immunogenicity as expressed by the interferon gamma responses. This was at the time that people were convinced that if you look at interferon gamma response, so basically T-cell activity, you'll get an idea about how immunogenic these vaccines are. And you can see here, this combination, BCG, MVA, 85A, by far the best. But then the phase 2B study was done in South Africa in infants. And it was reported in 2013, and it basically showed no clinical efficacy whatsoever. It was around 0. So this really set back the field because it showed that, first of all, that what we thought we knew from non-human primate studies didn't really translate into clinical efficacy. And the same was true for our interferon gamma response. So basically, we have no agreed laboratory correlate of protection. And this is very important because as long as we don't have that, trials need to be very large and of very long duration. So we basically don't know which candidates afford protection until we have tested them for clinical endpoints in phase 2B and phase 3 trials. And these are very high-risk trials. So they are high-cost trials, and for that they are very high risk, because we don't know if a candidate is likely going to succeed. The problem here also is that the key clinical endpoint for licensure of a TB vaccine for adolescents and adults is prevention of TB disease. So we want to make sure, in phase 3 trials, prevention of disease has to be the primary endpoint. But that requires very large numbers of participants, very large trials. They have to be followed up for a long period of time. That's basically just due to the natural course of disease. So duration of follow-up is generally 3 years or more. Now, there are several alternatives being tried in so-called proof of concept phase 2B trials with alternative endpoints. And the ones that are being used are POI, which stands for prevention of infection, and POR, which stands for prevention of recurrence. Now, POI trials basically look at conversion of intergamma release assays, IGRA conversion, as we used to have tuberculin skin test, nowadays IGRA, or sustained conversion. And in POR trials, one looks at recurrence to see whether after a patient has been cured, whether relapse or recurrence can be prevented by a vaccine. The problem with these endpoints is that we don't know how well these endpoints translate into protection of disease. So even if we have a phase 2B trial that shows a signal, for example, for prevention of infection, we are not yet sure that it will do well in a phase 3 trial with POD as the endpoint. So now currently, as I already said, we are in the stage where we can start trying to identify these correlates for protection, and also to validate these animal models, because now we are seeing, we do have trials that show protection signals. So this work is now actually only starting. Now, the fifth barrier, I think, is really the major one. That's funding. This comes from the Treatment Action Group report of last year, really a very important piece to read if you're interested in this field. Very thoroughly investigated. What you see here on the left is the 5-year research funding targets. And this is how well, so these are the white bars, and then the blue bars, greenish blue bars, they show you how far we are in the first 2 years. Now, and the two areas that are of relevance here are basic science and vaccines, and we see we are very far off target still. And a major problem here is the disbalance in funding. This is looking at funding for TB vaccines spent in 2019, a little over $100 million. $100 million. Now, I think that currently into COVID-19 vaccine discovery, I think spending has been on the order of 10 billion. So you can see what the huge difference this is for between these two diseases. But the real concern here is that is only two funders, the National Institutes of Health, US, and the Gates Foundation, that together account for 80% for all of the funding. So this is really a problem. And my big question is here, where are the BRICS countries? Where are the middle-income countries that basically have the largest share of all the TB patients, and definitely should have also the funding to engage in vaccine development and clinical evaluation? All right, so let's move on to our current TB vaccine clinical development pipeline. This is the pipeline, almost complete as recently presented by the WHO in their global report. The different colors, and you can see here, it goes from phase 1, 2A, 2B to phase 3. The different colors represent different types of vaccines. So the dark red are live attenuated vaccines, just as BCG. The ones that are sort of reddish here are whole cell vaccines, which basically means inactivated mycobacteria, or an extract of that. And then the green ones are adjuvanted protein vaccines, and the gray ones are viral vector vaccines. So these are the ones that we are now currently seeing a lot in COVID-19 vaccine development. So you can see that there are various platforms are being used, even though some of them are not too far yet in the pipeline. So what I'll concentrate on is the ones that I think are the front runners: M72, VPM1002 BCG revaccination, but also spend a little bit of attention to these 3 vaccines here that are also in phase 2B or 3. So let's start with M72. So this is a fusion protein of these 2 antigens, MTB39A and 32A, together with an adjuvant that was developed by GlaxoSmithKline. That's also basically the adjuvant in their pretty effective shingles vaccine. It's called ASO1E. This was published last year. This was the phase 2B trial, where they gave M72 as 2 doses versus placebo, 1-to-1 randomized in 3 countries in Africa with a follow-up of 36 months, and as the primary endpoint, bacteriologically confirmed pulmonary tuberculosis. About 3,500 participants randomized, and it showed a vaccine efficacy after 3 years of almost 50%. 49.7%, but as you can see here, with a pretty wide confidence interval. And this was really very good news, because after BCG, it was really the first vaccine that showed anything that looked like a sort of a serious protection signal. Now, the further clinical development looks like this. GSK doesn't have the ambition to move this product forward themselves, so they licensed it to the Gates Medical Research Institute, which now acts as the sponsor, for 2 trials. One is a phase 2A trial that's currently ongoing. It's about safety and immunogenicity in virally suppressed people living with HIV, around 400 in South Africa. And a phase 3 trial is currently in preparation that aims to answer two questions. One, does M72 protect individuals who are already infected with mycobacteria, so are IGRA positive, from disease, and then for how long? And the second question is, does it protect IGRA negative individuals from infection and potentially disease? It's currently being planned. They're probably looking at 14,000 or more subjects in very high incidence settings. And the trial will include both IGRA positive and IGRA negative individuals and people living with HIV, but primarily for safety endpoints. We'll talk a little bit more about that later. And what you can see on the bottom here is what is the current thinking about the timelines. The phase 2A trial, the safety and immunogenicity trial in HIV, is planned to report in 2022. The year after that, phase 3 will start. If it will be have a complete 5 years of follow-up, it will report only in 2031. Possibly it's going to be earlier, and then possibly with a licensure, or at least a conditional licensure, already before 2032. But it's still a long way to go. Now, the second frontrunner is VPM1002. This is a live recombinant BCG. And of course, here the question is about immunogenicity, is clearly safety. So various studies have been done. So the earlier trials were done in healthy adults, where it was safe and immunogenic, and then also in HIV-exposed and HIV-unexposed infants in South Africa. Only one small study has been published, only 36 patients, so there's not an awful lot one can say from that, but a larger study is about to be published. There are a couple of trials ongoing, and they are a bit sort of difficult to classify as being either 2 or 3. The first one is of prevention of TB infection in infants. So it's a POI endpoint. It looks at IGRA conversion. The other one is prevention of TB recurrence. So this is in patients who have been treated in order to see if the vaccine can prevent recurrence. And then there is a phase 3 currently being planned for prevention of TB disease, and that takes as the approach a household trial nested in a household context study. So the timelines here is that for the recurrence trial, one expects results in 2022, the year after for the infant trial that looks at infection endpoints, and then hopefully the phase 3 trial can be started by then. The third is BCG revaccination, and that is a bit of serendipity actually, because this was not meant in the original, this was not a goal in the original trial. So this is BCG given to adolescents who had been vaccinated at birth. And in the past, there had been studies looking at BCG revaccination. It didn't really show an effect. But this study sort of serendipitously did. So this was a trial that looked at phase 2B trial that looked at this vaccine here, H4IC31, versus BCG versus placebo in these adolescents that had been vaccinated at birth in South Africa. They were followed up for 30 months. And the primary endpoint here was quantiferin, so that's IGRA conversion, and they found no significant effect between the various study arms. But they did show that sustained IGRA conversion, which means that the IGRA remains positive also 3 or 6 months after, that actually showed a signal, and that was a vaccine efficacy of 45%, also again with a wide confidence interval, for comparing the BCG to the placebo, these two here. So that suggests that BCG revaccination actually might have additional effect, even though we don't know exactly what prevention of infection means in terms of prevention of disease. Still, it's been a reason for the Gates MRI to take this on in their BCG revax program. They are currently doing a phase 2B trial where they're basically trying to replicate this finding, again in South Africa, in 1,800 quantiferin negative adolescents, where as the primary endpoint is sustained quantiferin conversion, so it's initial conversion, and then still being IGRA positive 3 to 6 months after. But also look at exploratory endpoints, and especially to see if the correlates of protection can be identified. It started already in end of 2019. Its primary endpoint analysis anticipated in 2024. Now, I think most people find it very unlikely that there's going to be a large phase 3 POD clinical endpoint trial for this BCG revaccination. So one actually expects policy recommendation just based on the POI endpoint, probably somewhere 2025. Now, I mentioned these 3 other candidates. One is known as MIP or Immunovac. It is a heat-killed Mycobacterium indicus prani, and it came to the attention because it has been FDA approved as an immunotherapeutic and immunoprophylactic in leprosy. And earlier trials actually in TB have not been too promising. One of pericarditis didn't really show an effect. It did show an increase in HIV-associated cancers. A trial where they looked at recurrence. It was in retreatment patients. They looked at a sort of additional treatment effect. Didn't really show an increase in cure rate, and a trial of pulmonary TB has not yet been reported. But still, a phase 3 study is being planned for prevention of TB disease in exposed household contexts in India. So we're looking forward to seeing more about it, even though the earlier trials have not been too promising. A second one that was actually promising in the beginning, but is now a bit of a disappointment, is DAR 901. This is a whole cell killed Mycobacterium obuense. Phase 1 and phase 2A studies showed it to be safe and immunogenic. But just recently, a phase 2B trial was published that was done in Tanzania in adolescents that had been BCG vaccinated at birth but were IgRA negative and had quantiferon conversion or sustained quantiferon conversion as the endpoint, the clinical endpoint, so a POI endpoint trial. And it actually showed that after 36 months, for neither of these endpoints, there was any significant vaccine efficacy. As you can see here, this is really not a matter of sample size. It's just no effect whatsoever. So apparently this is not I don't think that this candidate is going to be carried any further. Then finally, one that is relatively far down the pipeline is H56IC31, which is a fusion protein of these 3 antigens here together with an adjuvant. That's for which they completed the phase 2-1 and 2A trials. And there is now an ongoing phase 2B trials looking at prevention of recurrence in South Africa and Tanzania. And actually here, the manufacturer, which is the Danish SSI, is actually planning to try and license this basically for this purpose, for prevention of recurrence. So this is where we currently are. The M72 and BCG revax and VPM1002 are potentially promising front runners, but it's going to take a while before we know. Now, a few words about recent developments in vaccine discovery. I think one very interesting one over the past years has been the success of a cytomegalovirus vectored vaccine. So that's basically Mycobacterium tuberculosis antigens inserted in a rhesus cytomegalovirus. They tried several viruses, and just sort of the overall story of what you can see here on this slide is that it was in non-human primates, it was tremendously effective. Some of the most effective combinations that one has seen, especially because it showed in 40% of animals, it showed complete neutralizing protection, which we've never seen, hardly ever seen with BCG. So this is something that's now being taken forward, and it's now in preclinical stage. Other areas of interest have been mucosal vaccination. There have been a number of studies of mucosal vaccination giving quite different results. But this study here that came from the Netherlands was one in which BCG was delivered to the lung mucosa by endobronchial instillation compared to intradermal administration. But they used a different system for which they think is more alike what happens in humans, a different system for challenging these animals with Mycobacterium tuberculosis, so-called repeated limiting dose challenge. So here the animals were vaccinated, either vaccinated or not, and then they were repeatedly challenged with very low doses. And as you can see here, This is this combination, BCG and MUC. It actually performs pretty well in the various readouts. And importantly, also here, a number of animals are completely protected. You can see here and here and here are completely protected, which hadn't been seen before. And the same was actually seen in this study, which I think was a real breakthrough, which showed that if BCG is administered intravenously, you can see tremendously strong effects here. And this was IV BCG. This is BCG given in various different ways. And you can see that with IV BCG, there is a sizable number of animals that had no sign of infection at all. So there was sterilizing, neutralizing protection. Again, what we haven't seen before. So apparently neutralizing protection can actually be achieved if, even with BCG, if it is given in a particular way. Obviously, the question is whether intravenous administration is ever going to work in humans, but this is now being looked into. Now, finally, a few words about implementation, because that is, I think, still an important bottleneck to look at. Even though we don't have the vaccines yet, obviously, we need to do the work. So there's a couple of things that to me stand out. First is the efficacy and safety in people living with HIV. Clearly, people living with HIV are a high priority target group for TB vaccination. And there are two potential issues here. First is that TB vaccines may have different safety profile in people living with HIV, particularly if they are live attenuated. And as I mentioned, also VPM 102 is live attenuated. TB vaccine candidates may show reduced immunogenicity and protection in people living with HIV. So that's the other problem. So the implications for this is that phase 3 trials of candidate vaccines should include people living with HIV, at least for safety endpoints, ideally also for efficacy endpoints. The problem is that this may be prohibitive in terms of cost and sample sizes if we don't have correlates of protection. So this is currently a big matter of debate. And obviously safety and effectiveness in people living with HIV need to be monitored closely after a vaccine has been introduced. A second issue is the problem of the target group. So if we are looking for a vaccine for adolescents and adults, it has to be used in these groups effectively. And we have very little experience of immunizing adolescents and adults, especially in low middle income countries, where almost, if not all vaccines are given at very young age. So the questions here are, do we need vaccination campaigns? Then at what age, at which intervals? What are its logistical implications? For example, how do we deal then with cold chain, route of administration? Community engagement is going to be tremendously important. We know that from other vaccines. We know that from especially from Europe and the United States, but that's probably going to happen elsewhere as well. What's called vaccine hesitancy. So these are really things that need to be addressed. And the implications are that we need real-life data on the requirements and best practices. And obviously, we can probably learn from the rollout that is now starting of SARS-CoV-2 vaccines, which are also given at these age groups. Then finally is that there is still limited political commitment and funding for implementing a new TB vaccine. And the issues here are that we don't know yet how the expected public health impact of a new vaccine compares to that of alternative interventions, for example, of tuberculosis preventive treatment. And now that it's becoming more and more accessible. Another issue is the cost of vaccination. Clearly, industry is not picking this up. So who is going to pick it up? How are we going to do this in a world where this vaccine needs to be given in countries where there is limited financial incentive? What will be the expected market size? It's going to be tremendously important to get manufacturers on board. And will multilateral funding mechanisms adopt a new TB vaccine? That's also very important. For example, Gavi, if they don't adopt it, who's going to pay for it in low-income countries? And the implications are that we need political and community engagement, and we definitely need funding mechanisms that guarantee manufacturing and rollout. So not only discovery and clinical studies, but also post-marketing. So in conclusion, first, an effective vaccine, in particular to prevent adult pulmonary disease, will be important to eliminate TB. 2. Key barriers to developing new TB vaccines include, first, limited knowledge of the human protective immune response. Second, lack of animal models that predict protection in humans. Third, lack of agreed laboratory correlates of protection. Fourth, as a result of that, need for large trials of long duration. And fifth, limited funding for TB vaccine R&D. 3. M72 and VPM1002 candidates are entering phase 3 and may be licensed within 10 to 12 years. BCG revaccination may become a policy recommendation for high incidence settings, even though we don't expect a phase 3 trial there. 4. The upstream pipeline is rather narrow, but there are promising new platforms and routes of administration. And 5. Key issues around implementation of new TB vaccines include efficacy and safety in people living with HIV, logistics of immunizing adults and adolescents, and lastly, political commitment and public funding. And with that, I would like to thank you for your attention.